on. After the colorless phenol reacts with the colorless formaldehyde to make a colorless product, then that product gradually becomes colored. And it is not oxidation related because I've done it in a closed tube with nitrogen, and it doesn't require the presence of oxygen. I made one, I guess you'd say, mistake last time, in that I said you could divide adsorbents into two kinds of compounds, the polar adsorbents and the carbon type adsorbents. I made a mistake in the sense that there's a third type that I forgot to include at the time, and that is the hydrogen bonding type, which become very important with phenols but are not very much important with other kinds of compounds. And these would include the PVP, polyamide, nylon type things that are used in wine. So they become particularly important with regard to phenols in that case. And they behave somewhat differently than the other two groups in a way that we'll discuss momentarily. So we were talking on the page 5. We're now turned over to page 6, getting ready to talk about some of the countercurrent or chromatographic methods of phenol separation. And under adsorption chromatography, to recapitulate a little bit what we said, polar adsorbents like alumina or magnesol, you would adsorb the material. If you want to adsorb phenolic material, you would put them on the column in the most polar solvent that the least polar solvent that you can, the most pet ether-like, the most gasoline-like, the most hexane-like solvent you can, and then elute them by increasing the polarity of the eluting solvent, so that alkaline water, perhaps, or alkaline methanol water would be the most polar thing that you could use. It turns out, in fact, you can dissolve alumina and magnesol if you get very strongly either acidic or alkaline solvents, so that you have to watch out for actually getting the adsorbent into your receiving fractions, but nevertheless you can get most everything off. Trouble, though, with alumina in particular is it is rather alkaline itself. Now you can buy acid alumina and neutral alumina and various degrees of activity of alkaline alumina, but phenols, once adsorbed on alumina, tend to be held so firmly and probably changed so that it is very difficult to get them back. As a consequence, it hasn't been as useful as it would appear that it would be on the surface. Fluorosil is a lot less potent in this regard, and magnesol, both of these are magnesium aluminum silicates, and they're not so alkaline, and it is more possible to recover things from them. The hydrogen bonding materials, notice that you would put on the phenols in water, and then the sugars and so forth would tend to go right through because they would stick or stay in the water and not stick to the adsorbent. And then you dilute the phenolic materials with something that would displace hydrogen bonds, or at least a very good solvent. So methanol and particularly acetone or formamide as displacers of hydrogen bonding. Formamide, you realize, is the amide of formic acid, and as you can realize, that's a pretty good hydrogen bond displacer. It both donates and receives hydrogen bonds, and so that it's a very good solvent to displace phenols from a hydrogen bonded condition, as would be the case on PVP. In fact, if you wanted then to regenerate PVP for use in a winery, say, it might be a nice solvent. On the other hand, it isn't the most pleasant thing to have in a food product, so that perhaps acetone would be better. At least I wouldn't mind drinking a small amount of acetone. I would mind drinking a small amount of formamide. Of course, you might prefer not to even drink acetone, so there isn't at the moment a really good way to regenerate PVP without running some risk of contaminating following batches of wine. Nevertheless, it would show you how to do it. And as a corollary then, among phenols, the glycosides would generally hold less tightly, and the more strongly hydrogen bonding type phenols would hold the most strongly. So the increased number and strength of hydrogen bonds that the phenol can undergo would make it stick the hardest, and then the more water soluble and the less hydrogen bonding, like in a glycoside, would stick less tightly. So a completely methylated phenol shouldn't stick at all on a PVP or nylon column. The acidic phenol should, and then the water-soluble effect of the sugars would tend to also keep them from sticking very well. There are a number of adsorptive conditions from things like ion exchange resins, where phenols will stick to ion exchange resins under circumstances where it is certainly not ion exchange. In other words, the resin is behaving as a plastic, and this plastic has hydrogen bonding capability or solubility capability that will cause the phenols to stick to it, or at least be retained to some degree by it. And this is not, generally speaking, truly an ion exchange process. But it does mean that if you use ion exchange resins to treat wine, you're also going to modify the phenolic composition, very likely, at least unless the resin is saturated with the phenols from recycling and is not regenerated by the regenerating ion treatment. Activated carbon is used mainly in wine to remove brown colors from white wine and so on, and is kind of a last resort because it tends to absorb odorous compounds more readily than it does larger colored compounds. But if you have a seriously defective wine, it probably can be used to clean it up. In terms of studying the phenols and getting them back from carbon, we've already discussed briefly that something like bromobenzene in a solvent that's very potent as a solvent for the phenol would be the eluting agent. This is a general principle, in fact. Things adsorb because they collect in surfaces. And if you're adsorbing, you should do it from the least effective solvent. In other words, the less soluble the material you're trying to absorb, the better chance it'll collect in an interface. Whereas if it's very highly soluble in the system, then it's not too likely to adsorb. Now, partition chromatography is, I've skipped over gel filtration. It's a laboratory technique. It's useful, but it's fairly obvious. It's a sieving type technique. Nevertheless, hydrogen bonding must be considered, and it's difficult to get all the phenol back because it tends to bind in poorly replaceable ways rather than just diffuse in and diffuse out as it should. Partition chromatography is rather easily related to the discussion we briefly had of the distribution coefficient. And so what you would want to do is to arrange the system so that the stationary phase, partition chromatography works by having a particle of some sort that's an inert support coated with a stationary phase, and then these beads, or whatever they are, have channels between them for a mobile solvent. So you have a mobile solvent that is the moving phase, and the stationary solvent, which is held in place. Well, in the usual situation, the stationary solvent is the more aqueous, the mobile solvent is the less polar, and by carefully arranging an immiscible pair of solvents, you can then go from extractive information with distribution coefficients to chromatographic type use of partition chromatography. The main thing to be remembered is that if your distribution coefficient was 1, chances are your material would come off in the solvent front. And the reason being that even if the flowing volume was the same as the stationary volume of solvent, a distribution coefficient of 1 would give you relatively little fractionation, and the material would be coming off very quickly because the mobile phase competes well for dissolving the material. So generally you need a distribution coefficient that's considerably less than 1 in order to make this system work, whereas in, say, distribution in separatory funnels or something of this sort, 1 is a fairly good one to use, or something near 1. And if you had something way less than 1, you'd be extracting the rest of your life to get things out. Even more is this a problem, since the typical situation, the stationary phase is a good deal less in volume than the mobile phase. So the mobile phase represents maybe a third, well, 2/3 of the volume of the tube that's packed, and then the solid support that has no particular effect anyway may represent a sizable fraction, surely will represent a sizable fraction of the remaining third, so that in fact the stationary phase is a lot less. So even more so, you must have your distribution coefficient considerably less than 1 to have the material stick on the column long enough to get any appreciable fractionation. And these same sort of considerations apply to gas chromatography, except the partitioning is between vapor and the stationary liquid, but basically the same sort of idea. And the phenol can be volatilized or made more volatile by silation, hanging on the trimethylsilyl units as ethers with the phenolic OHs and alcoholic OHs for that matter. This hasn't been, so gas chromatography being such a nice technique, why aren't phenols commonly analyzed that way? Well, there are a number of papers saying you can do it. Works rather well with pure phenols and model systems, but when you start applying it to very complicated natural mixtures, you have so much trouble getting complete silation. So if you had, say, catechin with counting the alcoholic, you have 5 OH groups to silate. Well, you could get some with maybe none, some with 1, some with 2, some with 3, some with 4, and some with 5, and you have very great difficulty getting all 5 completely silated, so you get a single peak from catechin. And if you don't, you might get 5 or 6 peaks, you see, from catechin, and then that makes it kind of hard to interpret. So that in a natural mixture, it hasn't turned out to be terribly useful so far, and I'm of the opinion it will not turn out to be so useful. Electrophoresis can be used, but if you're going to ionize a phenol, you'd have to do it in strong alkali, and this makes it unstable, as we've already mentioned several times. But a trick you can use is to use the borate complex, and an example is shown on page 7, so that if alcohol or phenolic OH groups are on adjacent carbons, vicinal OH groups will be complexed with borate to give a strong borate negative ion, which behaves as a strong acid and then can be used in electrophoresis reasonably well, and is also fairly stable. The pH need not be terribly alkaline, and of course it does stabilize vicinal OHs, which are the kind of phenols that tend to oxidize readily anyway. Having separated the material, you need to identify it, and paper chromatography and that sort of thing, column chromatography, can go a long way toward identifying the phenols, but you may also be interested in degradative type studies. And the two that ought to be mentioned are partial hydrolysis and the alkaline degradation to give the A and B ring parts separately. Demethylation and other derivatives are also mentioned. The partial hydrolysis, as you can visualize, is a way of showing particularly how many glucoside or how many sugars are attached to the anthocyan or to the phenol, and what order. So, for example, if you had a phenol that had glucose rhamnose glycoside, the glucose next to the phenol and the rhamnose attached to the glucose, then if you hydrolyze that carefully, you would first hydrolyze off the rhamnose, or at least some of it would hydrolyze that way, and then next you'd hydrolyze off the glucose. And if you did a paper chromatogram, say, you'd get 3 colored spots: the unchanged material, the one with the glucose attached and the rhamnose removed, and the one with both sugars removed. Whereas if, and then you could separate these and identify which sugar it was that stayed attached and which sugar came off first. So it would tell you the order of attachment. And you'd have the reverse situation if the rhamnose was attached directly. And then suppose you had the rhamnose and glucose attached to different phenolic OH groups, as your notes show, then you'd expect to get 4 colored spots. You'd get the monoglucose, the monorhamnose, the original sugar linked, and the aglycone. So in the first 2 cases you get 3 red spots, and the second case you'd get, or the third case you'd get 4 red spots, so that you'd be able to tell right off the bat how many glycosidic units were being hydrolyzed and in what order by this kind of a study. On the degradative reaction, it's a fairly obvious type reaction, and it's a lot easier to explain than it is to do it right. But by using hydrolysis in barium hydroxide in a sealed tube under pressure, or various such carefully done alkaline hydrolyses, if you have a flavonoid of any sort, there's our old friend again, you will get basically 2 units, and generally we haven't done much about this unit. In other words, it's not even very well studied whether you get a fluoroglucinol derivative, you do in part, or whether you'll break this down to further other products. But the part from the B ring in particular will yield generally the benzoic acid derivative, and in this case protocatechuic acid. So if you did a degradation of this type and studied the benzoic acid derivative that resulted, you'd be able to say what the substitution was on the B ring, because all of these benzoic acids are available for comparison. And you'd get the same B ring product from cyanidin, catechin, quercetin, anything else with the 3', 4' type substitution on the B ring. So this can be a very helpful way of studying the ratios or the identity of a compound of this type. And I've been quite interested in adapting it to study the ratios in a mixture, but so far haven't yet completed the job, and maybe some more years before we do. At least it might be possible to use this technique to get the ratios of malvidin to delphinidin to cyanidin, etc. in pigment mixtures, which at the moment is much more difficult than it might be this way. If you have questions, please stop me, because I realize I'm being kind of breathless about some of these things, and the main thing I'm trying to get across is the concept, so that you won't be completely startled by hearing about some of these things. Having had the phenolics class, you should know that these reactions exist. This brings us to paper and thin layer chromatography, having seen no hands waving for questions. And these we won't spend much time on, except to say that your notes are reasonably clear and, I hope, and explicit. Partition and adsorption chromatography are commonly used on paper. And how do you distinguish between the two? Well, typically in a 2-dimensional paper chromatogram, we would have one point as the starting point, and as you no doubt know, you'd run it in one solvent so that we get a series of separations along this axis with whatever solvent. I would commonly use BAW, which is butanol, acetic acid, water, in the ratio of 4 volumes to 1 volume to 5 volumes. So that's normal butanol, acetic acid, and distilled water in those ratios. So I'd commonly do that first. And then I'd run in the second dimension 2% acetic acid in water. And each of these would then migrate some way in the other solvent, so that we would no longer have that, and we would have spread them in the other direction, and you get a 2-dimensional mapping effect. Now, the main point I want to make is that the butanol acetic water is operating as a partition mechanism. The reason it is that you hang the paper in the chamber and let it equilibrate until it has absorbed from the atmosphere in the chamber the aqueous phase. If you don't do that, you can just start running and it'll absorb the aqueous phase out of this system. But then that'll mean you might get double fronting. In other words, you'll have some of the solvent will be dried. So what you should do is hang the paper in the chamber for a half hour or more, and it will take up from the saturated atmosphere the water layer on the paper. And then as you run the butanol layer, well, we're talking about running it this way. As you run the butanol layer, you'll be partitioning between the butanol-rich phase and the aqueous-rich phase, which is held tightly to the paper. So it's a partition system. On the other hand, in a water system that's 98% water and 2% acetic, then that's going to be an adsorptive system, and you're depending on the cellulose of the paper as an adsorbing agent. And that's one reason why this kind of a 2-dimensional system works rather well, because on the one axis you have a different mechanism of operation than on the other axis. If you did butanol acetic water in this direction and ethyl acetate acetic water in the other direction, then you'd probably just get a 45-degree line down the middle, which would be roughly the equivalent of running butanol acetic water in 2 dimensions, and outside of maybe being the equivalent of a longer piece of paper, you haven't really accomplished anything. So by having systems that work on different mechanisms, you get a much better type of separation. And on page 10, we say something about the interrelationships in this regard. Under item B, it's commented that the OH groups can be more or less counted up and in relation to the mass. So if the molecular weight is 300 and it's got 10 OH groups, 5 on a sugar and 5 on the flavonoid, then it has a very polar type condition, which means in the butanol acetic water it's going to be very water loving, so it'll have a low RF, a low movement ratio in proportion to the solvent in the butanol acetic acid water. So that if it has more OH groups, including sugars, it will, sugar or alcoholic OH groups, it will move more slowly in the partition type, butanol type solvents. However, in the adsorptive direction on cellulose, cellulose is a pretty good hydrogen bonder, and that's why it serves as a good fractionator of phenols, and only the phenolic OH slow the movement on the paper, and the alcoholic sugar type OHs will hasten it because they'll make it more water soluble and they're not acidic enough to hydrogen bond appreciably. So that what would happen if we had, say we had 2 substances that moved, they both have 10 OH, well, that's a little hard to say, maybe they talk about 7 OH groups maybe, 5 on the sugar and 2 on the phenol, and we then hydrolyzed off the sugar, so we're comparing the aglycone and the glycoside. If the ratios were about the same on a molecular weight basis, they might move on top of each other in the BAW, because as we remove the molecular weight of the sugar, we'd be dropping down to 200, and we'd have 3 for 200 compared to 7 for 300. It's possible that they'd move in the same RF value in the butanol acetic water. But in the acetic acid type solvent, where adsorption is important, then the aglycone with 3 phenolic OH groups would move less than the glycoside, which would have high sugar content and therefore be moved near the aqueous front of the 2% acetic. So that generally speaking, aglycones move little on cellulose if this irrigating solvent is a adsorptive single phase type material like 2% acetic acid in water. So if one wanted to, and I've been known to do this on midterms, which we will delay our midterms somewhat, but I might say here are these 2 compounds which would move most rapidly in these solvents on paper. Well, that may sound like you don't know, but you do know if you use this sort of rule. And the same kind of consideration can be used, as we just said, on a PVP column, for example, in that it's, again, it's a hydrogen bonding mechanism, so the phenolic OHs would stick to the PVP, and the sugar OHs would tend to hasten its movement and hurry it through, stick less well. Having prepared a paper chromatogram, or a thin layer, or for that matter, maybe a column chromatogram, you might want to identify the local occurrence of the phenolic materials after you're through. It doesn't do much good to separate them unless you can see them. Anthocyanins, of course, you could see immediately, but most of the other phenols would be too dilute or colorless, and you couldn't see them. Well, we've given you examples of several color reagents, spray reagents, and the generally most effective system to start with is to use UV fluorescence on the dry paper and locate the spots then. Maybe fume the paper with ammonia, because then the ammonia fumes will evaporate again and you haven't spoiled the paper. The ammonia fumes being alkaline will cause some of the materials to fluoresce better than they did without the ammonia, and sometimes the color will change. So commonly that's what we do. We view the papers under UV lamp. then a second time marking anything that fluoresces, a second time with UV and ammonia fumes, and then let ammonia evaporate and spray with the ferric chloride ferrocyanide reagent, which is mentioned as number 2 on part 5 on page 10, 5A2. This is probably the best general reagent. It does not react with all monohydroxyphenols. Some it will not react with. If you are worried about this, you can always use the Folin-Chicolteau reagent, for example, but it's much less sensitive. And one of the reasons this fresh mixture of ferric chloride and ferrocyanide is very useful is it's highly sensitive. About 5 micrograms of most phenols will show on that reaction, whereas if you use diazonium salt coupling, next 4 down the list, this will take about 10 times as much, 50 micrograms or so. Of course, it has the advantage of getting different colors, whereas the ferrocyanide mixture only gives blue spots with everything that reacts, including ascorbic acid and a few things that aren't phenols, whereas the diazonium salts will give not only more limited reactions, but also different colors with different phenols. And this can be quite useful. And then we have a few specific color reagents mentioned that are specific for certain structures that might turn out to be useful if you're looking for a certain one. The spectral data, any comments or questions on paper chromatography? All right, the spectral data I'm going to be even more cursory with, and that's, sorry about that, but I do feel we ought to mention it. Your notes are fairly good, I think, if you can decipher them. Some of the terms may be somewhat cryptic to you, and I think the best way to handle that is if you do have questions now or later, I'll try to answer them either individually or in class. The main point is that infrared, mass spectra, NMR, and fluorescence can all be very useful in studying and identifying phenols. But UV visible spectra are by far the most useful and have been most used. The basic idea is that as you irradiate substances with UV light of sufficient wavelength that it can interact, sufficient energy that it can interact with the molecules, then this energy is absorbed and converts the molecule to an activated state. Usually the kind of thing we're generally talking about is what's called a pi to pi star transition. Well, I'm not entirely sure I understand that. For that matter, I'm not so sure the people that tried to explain it to me understood it. But anyway, the idea is that you have raised some of the electrons to a more excited condition, and these electrons have taken up the energy of the irradiating light, and therefore have diminished the energy of the light, and therefore you can measure a subtraction. So you shine the light of a certain wavelength through the solution. Some of the energy is subtracted from it by the absorption and the raising of the molecule to the higher energy level, and you can measure the difference in terms of the light intensity. It means, of course, that a certain wavelength of light is necessary because it must interact with the molecular structure. And that, of course, is one of the major criteria. In other words, what is the wavelength of the light that is absorbed by a certain compound? The second consideration is the extinction coefficient, or the degree of intensity that the light is absorbed. So a certain wavelength is required, and then a certain efficiency in using that light is related to the molar extinction coefficient of the compound involved. Now, the phenols generally absorb, as we've already indicated, about 280 nanometers. And if you compare that with benzene has an absorption, a rather complicated absorption, but the longest wavelength band is about 254. So putting an OH group on benzene raises the extinction coefficient about to 280, and therefore you can tell a benzene from a phenol by this kind of increased length. And most phenols tend to be in about the same position. And then if you ionize from the phenol to the phenolate ion, in other words, place the phenol in the alkaline solution, then you'd ordinarily get a further increase of the wavelength from 40 to 100 nanometers. So 40 would be 320 and 100 would be 380. So we generally expect that in the phenolate condition it would absorb at considerably increased wavelength. And you notice 380 is on the borderline of visible, 400, and so it would certainly show yellow. So this compound would be colorless, you couldn't see it. Add it to alkaline condition, and if you got a 100 nanometer shift, it would become light yellow as you overlapped into the visible region. So in alkaline solution, the phenols may be colored, and if they're already colored, they're going to be more colored generally than they were as phenols. The shift to longer wavelength is called a bathochromic shift. And that's also a red shift because you're getting a longer wavelength. The infrared is on the long end and the ultraviolet is on the short end, so you can remember that. So a bathochromic shift is a red shift, and a blue shift would be a hypsochromic. And the way I remember that, you won't like me saying it probably. Most classes have shuddered when I say this, but the way I remember this is that hippies tend to be blue, but if you make them take a bath, they turn red. So remember it that way if you want. In any case, remember it however you want. This is one way to keep the thing in mind. And it is true we're dropping the bathochromic and hypsochromic nomenclature, I think, for red shift and blue shift, which may be more sensible anyway. And at the top of the next page, which I think is unnumbered, but it'd be 13, I believe, we have examples of the two major bands in the UV visible region for a series of types of phenolic materials. And we've labeled them band 1 and band 2. Now it turns out that band 1 is generally reflective of the B ring substitution, and apparent in a flavonoid at least. Now, of course, you see it's present in the synamates, which don't have a B ring, A ring composition. But the reason seems to be that you get this kind of equilibrium, and maybe I better look at my notes, make sure I get it straight. So that we can have this kind of an ionization. Well, let's draw it this way. So we can have this kind of an ionization which involves the B ring, and the B ring involvement, in other words, substitutions on the B ring, then will tend to affect the band 1. So band 1 tends to be related to substitution in the B ring, whereas band 2 tends to be related to substitution in the A ring because it seems to be involved with this kind of ionization. And we can put the charge on wherever we like, but we put it on in the other place there, so we could do it that way this time. So that the A ring substitution is going to affect this kind of so if you put an OH on here, it's going to affect this ionization situation much more than this, because this unit will behave more or less as an isolated fluoroglucinol ring, and one more OH, fluoroglucinol tends to absorb about the same as resorcinol, and that's about the same as phenol. So substitution here wouldn't affect this situation much, but it would affect this situation, and vice versa. So that the two bands and how they change gives you a pretty good clue as to the nature of the flavonoid you're talking about and what kind of substitutions are involved. And without trying to make any great study of this, I at least wanted you to know that it was possible then to learn a good deal about phenols by studying the UV invisible absorption spectrum. Phenol substituents, that is, substituents attached to the phenol, tend to give sizable and predictable kinds of shifts. For instance, another OH group is what's called an oxochrome. In other words, it adds to the color, and it can give zero shift, but it tends to give a shift toward the red wavelengths. And remember, we said in benzene, if you add an OH, you go from 254 to 280, so you're getting a red shift there. And that same kind of thing is carried on if you put in a second OH on a phenol. So ordinarily you do get a red shift if it's one that interacts.'However, as I just said with fluoroglucinol, it need not always do so. So for example, if you have a flavonoid with a 4' OH group, the 4' OH compared to a bare B ring would make quite a difference. But if you add the 3' group instead of the 4' group, it doesn't have much effect. However, if you have the two together, then again it has an effect. So you have to know a good deal more arbitrary things about the molecule, but ordinarily a good deal of information can be gleaned by comparison with the literature. Of course, any sort of a conjugated double bond will shift the wavelength toward the visible or a red shift. And for example, if you compare caffeic acid with the hydrogenated version, the double bond in the cinnamic acid side chain makes quite a difference. Dihydrocaffeic, as your notes say, is 280 nanometers like any other phenol, whereas caffeic with the double bond present and in conjugation with the carbonyl of the carboxyl is 324 nanometers, quite a sizable shift. And there are other descriptions, whereas the hypsochromic, or the groups that tend to shift toward the shorter wavelengths, would include methylation and acetylation. So if you want to study a phenol, you can make the methyl and acetyl derivatives, and the acetyl derivative tends to look like the parent hydrocarbon should in UV absorbance or visible, and the plain phenol will look presumably different depending on the number and placement of OH groups. And there are a number of shifts that can be tabulated, and both on your notes and the final page taken from Mabry's book, we have some descriptions of typical reagents to cause shifts, and these shifts have been tabulated by Mabry and his co-workers in the book reference given, so that if you want to compare with a known compound, you can. They're all based on complexes or ionization effects, so that if you have a very acidic phenol, it may ionize in sodium acetate. If it does, why, it'll give a red shift of some magnitude. Any of them will ionize in sodium ethylate and will give a phenolic red shift, a phenolate red shift, and so forth. So, and the boric acid complexing will influence this, and so on, so that a great deal of information can be determined on an isolated phenol from the spectral data. Well, I think that completes everything that I felt I had to say about these laboratory notes. Any complaints, comments, rebuttals, whatever? All right, then let's go on to then start talking about the plant physiology. And again, I'll try to minimize our discussion here with the idea of bringing in as much applied information as seems reasonable, and yet trying to cover the basic material. The cytological aspect, there are a number of general references given, and although that particular list isn't as up-to-date as some, before, I think on the last page of these notes, there are some quite recent papers that, in fact, I read some of them last night, just got the book day before yesterday, always seem to be just barely staying ahead of the class. But in any case, these newer references do cover some additional information which doesn't modify the earlier information we're giving you. We've mentioned that the phenols tend to be synthesized in the cells where they occur. Well, and there are discussions here of some suggestion as to how this happens. The best and most complete information that adds to what your notes say is from a paper by Helen Stafford in this Recent Advances Volume 8, which just came out. And Helen Stafford is at Reed College or University, whatever it's called, in Washington, I believe, at least up the coast. And she has been doing very good studies in trying to integrate the enzymatic production of lignin with the physiology of the plant. And this is her picture at the present time, which comes as near as anything. Most of her paper, if you read it, is talking about the enzymatic activities rather than the cytological location. But she does have this one picture, which is kind of useful, and I'll go through it so you can understand it. This endoplasmic reticulum is a structural, microscopically identifiable tissue in the plant cell, and it apparently produces vesicles, and these vesicles apparently can break loose from it and migrate in the cytoplasm. And so the endoplasmic reticulum vesicles are just little tiny vacuoles that form and break away. They're not air-filled now. They're not like balloons. They're liquid-filled, membrane-bound, presumably. But they break away, apparently are formed and break away from the endoplasmic reticulum. And as your notes say, other people have said that these are, in fact, such things as amyloplast, which is a fancy name for a starch generating a starch granule bag, if you want to put it that way, so that the phenols tend to be formed, at least according to some information, from a starch unit that is migratable within the cell. Now, she doesn't go into that, and there's no clear evidence right now. But anyway, whatever the form of these vesicles, they do apparently take up phenylalanine and products of the shikimate pathway and contain within them such things as paracoumaric acid, caffeic acid, ferulic acid, and synapic acid as the cinnamic acid analogs that are going to be turned into other phenolic derivatives. And these other phenolic derivatives are apparently produced by interaction of these migrating vesicles with other particulate matter such as plastids, mitochondria, or other microbodies within the cell. And this is pretty uncertain, and you might argue about how do two organized elements interact, and she spends some time discussing this, at least arguing that it is possible and giving examples. Certainly as they touch, their membranes could interact in transfer manners or affecting reactions at the surface. In any case, these vesicles apparently pick up or produce or collect in some way the caffeic type derivatives, and then they will react with such things as uridine diphosphoglucose to give a glucose ester, or of course a glycoside is possible too, but in the cinnamic series it generally would be a glucose ester, and presumably the same sort of thing with quinic acid. So the esters are apparently formed at one of these particulate bodies, and then the vesicle with the esters instead of the acids migrates to the vacuole and breaks into the vacuole so that the chlorogenic acid type derivatives collect in the vacuole and are only present in the cytoplasm as a transient condition in these small vesicles. And this, of course, would keep them separated from the enzymes which are in the cytoplasmic tissue and not in the vacuole. And that must, this definitely does occur, at least on a concentration basis. Most of the chlorogenic acid would be in the vacuole. And the same is true of the flavonoids, which would be the same way, except that they would involve malonyl-CoA and CoA formation. CoA from the, make the ester derivative, I mean the acid would be a CoA ester, and then link the malonyl-CoA and form the flavonoid, and again the vesicle breaking into the vacuole. There is some reasonably good evidence, and this evidence is perhaps best in a paper by Shandarel, who is the father of Sig Shandarel at Fresno, CSU Fresno, and he has a very good paper on the microscopic evidence within grape tissue, which is the best of any plant study made yet, I think, showing these sorts of vesicles breaking into the vacuoles and showing that they can be of more than one kind, showing that some can contain tannins, apparently, are brown, and some contain anthocyanins and are red, so that, and they don't seem to be mixed. You make anthocyanins in one and tannins in the other, according to the best kind of information. That tends to be true of the vacuole, but is less so. You do find tannin and anthocyanin in the same vacuole, but if you find an extremely high content of one, you tend to find a low content of the other. So you will find cells that are very high in tannin and contain no anthocyanin, and or very little, or vice versa. Well, to continue the picture, another function of these migrating vesicles is to contribute to the lignin in the cell wall. And this apparently happens by making, again, the CoA ester of the phenolic acid, and then the reducing enzyme that converts the carboxyl group to the alcohol group acts, and just where we don't know at this point, but it is in some particulate function away from the wall itself apparently, but near the wall. And then the peroxidase is formed in a similar vesicle, and the hydrogen peroxide that oxidizes in the presence of the peroxide this alcohol to the lignin. Apparently these get together at the wall, and you do form then the lignin outside the cell in some extruding type mechanism apparently that again is not very well understood. It is pretty well known now that the formation of lignin involves peroxidase oxidation of these reduced compounds, so that that part seems quite clear. Well, that's not entirely satisfactory, but that seems to be about the best we can do in terms of explaining physiologically and structurally what goes on within the cell. And then chemically, I think we already know pretty much what goes on. In other words, as indicated, you'd be going from the cinnamate type derivative to the flavonoid, to the ester, to the glycoside. And these would be the substances within the vacuole. The translocation, even within the cell, is apparently related to either this vesicle type transport or a glycosidation or other solubilization effect. So as the phenol enters into the vacuole or before, it is apparently converted to the glycoside. So within the vacuole, you generally get only the glycosidic phenols or the chlorogenic acid type unit, which is also solubilized by the quinic acid ester. This can be shown, for example, in xanthium, which I believe is cocklebur. I'm not entirely sure, but anyway, the xanthium has been studied, and the agent used was 3,5-dicafeoyl quinate. Excuse me, the agent used was chlorogenic acid, and if it was fed external to cells of the xanthium, then as it went through the cell wall, it was converted to lignin. But if it escaped, radioactive mark on it, of course, if it escaped lignification as it passed through the cell wall, then it became dicafeoyl quinate inside the vacuole. So that apparently this esterification change is related to some of these protective reactions. In other words, in one form it's effective, in the other form it's prevented from acting, and this seems to be generally the situation. We mentioned earlier the IDO Blast, which are cells that are so full of tannin that they look as though they're completely destroyed. That's what the blast would mean, self-destroyed, IDO Blast. And in a typical tissue where these would be the plant cells, an idioblast is likely to be larger than several cells taken together. So the idioblasts tend to be in large cells. They look sort of like storage bags, dead cells, and so on. But some fairly recent work, which is reported in your notes by Constable, shows that these do revert, at least their evidence seems quite good, that they can revert, they are still alive, they can revert to a less tannin-filled condition which will undergo mitosis and divide into meristematic cells. And this has several important relationships. One, it shows there is a mechanism to solubilize the deposited tannin, and two, it shows that the cells are not in fact dead, as you might have assumed, considering their large and distorted shapes. The location of these kinds of tannin-rich cells, and they aren't all idioblasts in the sense of being super enlarged and distorted in appearance. Some of them are fairly normal appearing cells, but have a very high content of tannin, normal in shape and size. These tend to be located around, and sometimes the idioblast too, around vascular tissue, so that if you have a vascular strand, a vascular bundle, you tend to find a high concentration of tannin cells around it. They tend to be located at mobile cells, like in the mimosa, the sensitive plant that grows wild in Hawaii, and I always love to touch because it would fold up as soon as you touched it. This sort of action seems to be related to tannin content, and the motor cells, the ones that do this movement, are very high in localized tannin, apparently. Well, the other class is getting nervous, and I guess I must